Chapter 6 starts introducing us to the cell itself in more of a generic term, different aspects of cells. This is kind of an artist's rendition, makes it very simple, might be one that you end up referring back to. On the left you have we know it's representing a plant cell because it has that cell wall that is unique to the plant cell, that structured cell wall. And on the right, you have an animal cell. Both of these are eukaryotic cells because their organelles are membrane bound. We talk a little bit more about that. And then the other thing that this one's doing, again, that makes it a little easier to organize it in your head is those organelles that are unique to plants. It's listed them in green: chloroplasts, cell walls, and plasmatatas, and centrosomes, lysosomes, and centrioles. Actually, that's plasmodesmata. Sorry about the pronunciation of that. In terms of how we know that these things are in plant cells and these things are in animal cells, we use a microscope. You'll use a microscope in class, and the study of plants and the study of cells, or those things that are too small to see with the naked eye, is called microscopy. And that has been done over time. Certainly started way back in 1500s, and has been refined until they're still refining microscopes now, making them more and more powerful, more and more specific in what their function is. So the first study of the first microscope studies are attributed in terms of the cell wall and seeing differentiations to Robert Hooke. And he looked through microscopes to see dead cells. Microscopes then evolved into what we consider light microscopes. And the positive aspect of light microscopes is you have a light passing through a specimen, and it uses a lens to bend the light in such a way that the specimen is magnified. Light microscopes have three basic positive properties: magnification, resolution, and contrast. Magnification can sometimes be 1,000 times the size of the actual specimen, depending on how strong the microscope is. And even with that 1,000 times magnification, the resolution can remain. And then contrast in the fact that you, most of the time when you're looking at a cell, you are going to stain it, and that stain will help bring out the contrast, but sometimes it's important to look at a non-stained to see the cell in relationship to the organelles, one in relationship to the other. And so with the non-stained slides, the contrast is important with a light microscope. Other types of microscopes use electrons. Electrons are bombarded through the slide, and we have electron microscopes. They're more powerful. They can magnify to a greater degree, keeping the resolution and the contrast. And of electron microscopes, there are a couple of specializations. You have scanning electron microscopes, which we call SEMs, and what they do is they bombard the surface of what one is looking at, and that makes great topography so that you can see what the outer surface, what the outer surface would look like texturally, actually, to tell you the truth. And then you have transmission electron microscopes. What they do is they cut the slides into little slices. So, and the electrons are bombarded through, so you have the ability to, so one has the ability to study the internal workings of these organelles. So this is pretty fabulous in terms of understanding on a more nano, I guess in terms of nanometers, measuring things in nanometers to understand the very, very minute internal workings of the organelles of the cell. And you have, in this image, you have starting at unaided eye, how deep you can see. Using a microscope, what you can see is the animal and plant cells, nucleus, bacteria, mitochondria, and then electron microscope, how much more powerful they become. You get to the point where you can actually see small molecules into what is considered atoms. So you can actually see proteins and how proteins are formed. Viruses, being much smaller than bacteria, can see those with electron microscopes. And then this is a nice blow-up of what can be seen with a scanning electron microscope. The image at the top, remember that's what I was talking about the topical surface, the topography. You have the cilia on a, this could be a respiratory cell, and then transmission electron microscopes, where you're looking at a cross-section of the cilia, so you can see the actual design of the myofibrils. The cilia are made up of myofibrils, and that design of the how that design is created. One of the ways that we isolate organelles for study is called cell fractionation. And what happens is we'll put cells in a centrifuge. So you'll homogenize them, so kind of like what you think is take the tissue, homogenize it, so kind of put it in a blender is what you can think about. And then so you're breaking it all apart. So you're breaking the cell membrane apart. You're breaking it into parts of things that may weigh more than others. So what we try to do is get the heavier things to fall down to the bottom of the test tube. So that homogenate is then going to be centrifuged, and a centrifuge is kind of like one of those whirly rides where you're kind of thrown to the back of your seat in the whirly rides. And what happens here in the centrifuge, the heavier things are thrown to the bottom of the test tube, and what happens is it differentiates the items that are found in the cell by their levels, and can be then taken off, can be pipetted off based on their levels and put on a slide and then studied. So the parts of the cell can be fractionated away from one another and then studied. Biochemistry is and cytology help correlate cell fraction with structure. So once it's looked at and you can see what it looks like, biochemistry then can come in and say, well, this is why it looks that way, because this is the function of that particular organelle. So what happens is this data is not only observed by biologists, but also used to make inferences and hypotheses about what that structure has been created for. So it works kind of in symphony with all the other, with the many branches of biology. Eukaryotic cells have internal membranes, and you'll hear this over and over again. Eukaryotic cells have internal membranes. We have an external, you have an external membrane no matter what cell you are, but eukaryotic cells have an internal membrane that that compartmentalizes that organelle off from the rest of the cell, and that organelle off from other cells, other organelles that are found within the cell. There are two types of cells, prokaryotic and eukaryotic. And it's the eukaryotic that have these internal membranes. Prokaryotic tend to not have the internal membranes. And you can look at the bottom of the slide here and also in your textbook where you have a blow up of a prokaryotic cell. You're seeing a bacterium image in your textbook. That bacterium image is also viewed as a with a transmission microscope, where you can see the chromosome is not membrane bound, where in a eukaryotic cell the chromosome is bound within the nucleus. And only organisms of the domain bacteria and archaea consist of prokaryotic cells. And that tends to become a question to ask on quizzes or tests. Protists, fungi, animal cells, and plants all consist of eukaryotic cells. So the organization, you have two basic types of cells, eukaryotic, prokaryotic. And it's the eukaryotic that have membrane-bound organelles. Those are your protists, your fungi, animal cells, and plant cells all fall in the eukaryotic category. Bacteria and archaea are in the prokaryotic category. Some features of cells: you have your membrane, your plasma membrane, so your cell membrane. You have a fluid substance within the cell, which we call the cytosol. You have chromosomes, because we have to have some sort of way to express our information, if we are a cell, whether we're bacteria, whether we're fungi, whether we're animal, off to our offspring. So we have to have a way to reproduce ourselves. And that reproduction has to be organized in some sort of information fashion, which is chromosomes. And then ribosomes to make proteins, because in order to have any kind of metabolic functions, that's then is directed by our genes, but our genes can't leave the cell. What can happen is we can direct a protein to be made, and that protein then can leave the cell. So we can respond to our environment. We can respond to other cells that are similar to ourselves, but we do this via a chemical way, and that chemical way is proteins. Prokaryotic cells, again, it bears repeating because this is important information. Having no nucleus, DNA is in an unbound region. Now, there's no nucleus, but the DNA kind of stays in one area called a nucleoid. So it's kind of a precursor to a nucleus. It doesn't just float around. There are no membrane-bound organelles, but the cytoplasm is bound by a plasma membrane. So this is a unique cell. It has its own unique space. Eukaryotic cells, characterized by having DNA in a nucleus that's bound by a membrane, and generally we call it a nuclear envelope. One of the reasons we call it a nuclear envelope, not just a membranous or a plasma membrane, or something similar to the term plasma membrane, is because things can actually exit. You have little pores, little openings that allow eventually RNA to exit. RNA is the helper of DNA. You have membrane bound organelles that are each have their own unique function, and cytoplasm in the region between the plasma membrane and the nucleus. So you have a fluid environment. Prokaryotic cells are generally larger than, I said that wrong and I apologize. Eukaryotic cells are generally much larger than prokaryotic. This whole idea of having membrane-bound organelles have allowed eukaryotic cells to vastly increase their size is just a function. And we'll pick this up on the next teaching part.